Hi, everyone. I am Jack Clark, AssayQuant's product manager. Thank you for tuning in to watch our president, CEO, and CSO, Dr. Eric Schaefer, talk about the company that he co-founded, AssayQuant Technologies. Today, he will give a presentation titled Leveraging Continuous Fluorogenic Assays for Kinase and Phosphatase Activity to Enable Drug Development, where he will give a great description on AssayQuant's PhosphoSens technology and its continuous direct catalytic platform where you can see a progress curve in every well. The presentation is 35 minutes long, so if you need to put us on pause, please don't worry. We will be here when you get back. We would love to take your questions in real time, but obviously this is a recording, so we can't do that. Please write down any questions or thoughts you may have during the presentation and contact us directly at hello@assayquant.com. We look forward to hearing from you. The floor is now yours, Eric.
Great. Thank you so much, Jack, and for everybody participating in this call and viewing the presentation in the future. Indeed, it is an exciting time to be working on kinase and phosphatase drug development, where we are seeing tremendous growth in the diversity of drug chemistry and equally impressive results. To this pursuit, our proprietary PhosphoSens technology yields better, more reliable data, enabling better decisions about lead candidates and empowering faster movement through the drug discovery process. To do this, AssayQuant offers both catalog products. We sell simple-to-use kits, as well as bulk sensor peptide reagent. And we also offer services as either assay development services or custom compound testing services. Here is the PhosphoSens technology. These are biosensors for continuous monitoring of protein kinase activity. In this format, we take peptide substrates, which can be any commonly used or frequently, more often now, we're using only physiologically relevant sequences. And the only change can be insertion of a cysteine residue to which our Sox fluorophore is covalently attached. We insert this as part of solid phase peptide synthesis in a position proximal to a serine threonine or tyrosine phosphorylation site. So when you take this sensor peptide, incubate it with kinase, ATP, and magnesium forms a bridge with the phosphate group and the quinoline moiety, and this is what results in immediate increase in fluorescence so that one can monitor this in real time. So the assay format uses the sulfamidoxine or SOX chromophore, and the signal is generated by chelation-enhanced fluorescence. As I said, SOX is covalently attached to the cysteine residue, so it doesn't come off with reducing agent. And the configuration allows any amount of flanking sequences on either side of the phosphorylation site or the SOX fluorophore. The assay is direct, homogeneous, and kinetic, that is continuous. It's a simple add and read format, and you can walk away and do other things. It is ideal for quantitative assessments, including profiling, potency, and mechanism of action. And it's covered by multiple patents exclusively licensed to AssayQuant from MIT. The platform has been successfully validated by many companies and academic research institutions. Currently, we have over 368 protein kinase assays, and we are actively adding to this every week. We had initially many requests for a redshifted wavelength for our format, and we indeed tried various approaches to do this. It turns out to be actually much simpler than we had initially thought. So what one can do at any time during a kinetically run assay is to add europium. And so you can follow the curve initially kinetically, you then add europium, and you do an endpoint time dissolved fluorescence readout. So you don't want to do this kinetically anymore. You then switch to an endpoint format, and it is that europium immediately displaces the magnesium, resulting in higher affinity binding. It then gives you a time-resolved fluorescence readout, where you can read the emission at 616 to 620 nanometers. You can include 100 microsecond data acquisition delay, which together eliminates then compound interference. So this format is ideal for high numbers of tests for HTS runs or SAR. And this patent was just recently approved. Importantly, when you think about many of the redshifted fluorophores that are used commonly in various assays, they all become very large, hydrophobic, and can then present artifacts in terms of binding compounds or otherwise inappropriately modifying your assay format. As shown on the bottom, SOX is very small, and this was done by design to make it both minimally hydrophobic as well as small. So it's actually very similar to the size and hydrophobicity of tryptophan, which is shown further on the left. Again, one can simply add europium to convert from a kinetic readout to an endpoint red format, still with a very small and minimally hydrophobic fluorophore to minimize any artifacts. The PhosphoSens platform workflow is shown here. You can run these assays on any type of plate, 96, 384, or 1536 well plates. We've had many groups miniaturize the PhosphoSens red format down to 4 microliters in 1536 well plates to do high throughput screening, and we appropriately then can provide very competitive pricing to do this. You can read then these assays in any fluorescence microplate reader that's appropriately set up for either running kinetics or for doing the time-resolved fluorescent read. The assay, as I mentioned, is a simple add and read. And so again, shown in the blue ribbon is just the peptide substrate with the SOX fluorophore integrated proximal to the serine threonine tyrosine residue. You get phosphorylation, and you could then read either kinetically or as an endpoint red format. Again, endpoint red is really ideal for HDS and SAR. The kinetic format is ideal for profiling, potency assessments, mechanism of action, and biomarker. So in the kinetic format, as shown on the bottom on the right, you have a progress curve in every well. So you can see here in a 384 well plate, every reaction is monitored kinetically and typically for at least an hour, but we'll do reads up to 4 to 6 hours, depending upon the nature of the assay. You can do a lot of really great measurements in terms of kinome profiling, enzyme kinetics, or drug functional properties. And with the selective sensor peptide, can start to look at activities then in crude unfractionated cell or tissue lysates. So simple, powerful, and very flexible. And I'll talk about these applications in the subsequent slides. So the SOX fluorophore is very important, being small and minimally hydrophobic. But equally important is the peptide sequences that we have identified that really make then the selectivity for a given kinase of interest. So again, we can take any sequence. We will put systematically the cysteine SOX in various positions from -5 to +5 to test systematically where the SOX fluorophore is best tolerated. And we do this in a high throughput peptide format so that we can screen thousands of different peptides across many different targets to understand the properties that we can deliver with an appropriate sensor peptide. So currently we have greater than 30,000 CSox sensor substrates as a master panel with a focus on physiological sequences, and we can go to this and rapidly screen and create new assays. As highlighted on the left, we initially made many substrates that are commonly used, and many of these date back to P32 ATP assays where there are multiple lysines or multiple arginines included to be able to make the peptides better bind to P81 paper. We've systematically tested whether these basic residues actually change the functionality of the substrate, and indeed we've shown that systematically. So we avoid those sequences and focus on physiologically relevant sequences for our peptide sensor substrates. So the process we use for assay development is very well established. We do this under a master service agreement, and we've done over 100 projects successfully. So milestone 1 is to simply screen subsets of our 30,000 master panel. We key in based on what we understand about the kinase or based on an initial series of screening efforts to identify what that specificity might be. In many cases, we can identify in this milestone 1 a very robust assay, and within a week, we have generated an assay that can be used for drug development. If the assay is giving us initially a relatively weak signal, there are a series of optimizations that we can do that better present the peptide to the kinase of interest. And so milestone 2 is where we substitute every position with 16 different amino acids, with the exception of the phosphorylation site and the CSox fluorophore. We then test systematically whether any of these substitutions result in improved performance. And milestone 3, what we'll do is combine in all the different combinations any of the favorable substitutions identified in milestone 2. We then scale up the final peptide, and that becomes our final product. So just as an example, we can pick out any branch of the kinome, in this case, looking at the tyrosine kinases. We can then go through this optimization process, and we can pull out very generic sequences like AQT 101, 102, 104, and 794. That's shown in the upper panel, where we can see 80 to 90 different kinases with an individual sensor peptide substrate. So this is very good when groups want to minimize the number of sensor peptides that they stock, and can just pull these out and then routinely measure any number of different tyrosine kinase targets. There's also an increasing request for selective sensor peptide substrates that enable then unfractionated cell or tissue lysate analysis to look at activity of complexes then involving that particular target kinase. So the screen on the bottom then is going through the same optimization process, but then selecting for those sequences that give you more selective recognition. And so what we see here is a single peptide that works really well with KDR or VEGF receptor 2, but none of the other kinases give appreciable signals. So this is really the power with which we have harnessed high throughput peptide synthesis to really get at better substrates for kinases as well as phosphatases. So as an example, EGF receptor, very hot target. We have many groups that are working with us on this or related targets like HER2. And what we did here was to do an initial screen for milestone 1, where we pulled out a sequence that worked well, but we wanted to improve it. So we went through milestone 2, which is the round 1 on the left, and here you have 113 sequences that actually then differ by only one amino acid in each case. And these are progress curves that show you then the relative fluorescence over time. And so you can very readily see which sequences are actually giving you steeper progress curves, whereby we then use those individual substitutions and combine them in all of the different combinations for milestone 3 or round 2. And so what you see is some sequences, combinations don't work well at all. The green line at the bottom, that progress curve for an individual peptide. But obviously, we want to focus on those progress curves that show much steeper slopes. And so the orange at the top and those related sequences, what we'll do is take those and then do a scale up in HPLC purification to generate a final sensor peptide. In this case, we're showing on the third panel AQT 734. And so using this at 10 micromolar, we see really robust signals, so reaction rates that are greater than at least 1,400 RFU per picomole per minute, and some of them as high as almost 12,000. So they are very robust substrates. We can go down to very low nanomolar enzyme levels, and we can use the same sensor across multiple mutants as well as the wild type EGF receptor. We've done the same process for serine threonine kinases, and what I'm showing you here are just 5 different examples where different clients came to us saying, they're using protein substrates now, and they really wanted to then shift to a more drug discovery compatible peptide-based assay. And so in each case, we went through the 3 milestones, and we then can pull out and identify very robust sensor peptide-based assays in each of these cases. So it really opens up new regions of the kinome to rigorous drug development. And we've been able to do the same process where we identify improved substrates, sensor peptide substrates, that allow us to look at activators. So we have many groups coming to us saying they would like to develop activators of a particular kinase or phosphatase where that target is either downregulated or exhibits lower activity in a particular disease state. In each case, then, what we've been able to show is that we can identify sequences that give very high reaction rates, but allow very little activation. And there are other sequences that give high reaction rates, but allow significant activation with an appropriate ligand. And so that's what I'm showing you here is those sequences that are very compatible with activation for AMPK, DNAPK, PKCs, PKR, and CAMK2 alpha. So the same Sox-based format can be used for phosphatases. In this case, what one starts with is a phosphopeptide substrate. You then add the appropriate phosphatase, and you see immediate dephosphorylation, and then you follow loss of the signal in the same way, determining initial reaction rates and so forth. So same mechanism of chelation enhanced fluorescence for the kinetic assays, and you can use europium for the time-resolved fluorescence endpoint red format. Again, only difference is that we use CSox-based phosphopeptide substrates, and we've had many clients say to us that these assays are much more sensitive than when they've used small molecule phosphatase substrates, and it actually then allows them to better screen for differences in mechanism of action between their lead compounds. So data examples here are shown. Then on the left is using PTP1B with AQT266 sensor peptide. The blue progress curve shown that's very flat at the top is your control. So there's no phosphatase. And so you're just seeing the signal with the appropriate phosphopeptide. You add increasing amounts of phosphatase, and now you rapidly dephosphorylate the site of interest. And if you look to the right, you see really nice linearity that extends down to 20 to 75 picomolar of PTP1B. So very sensitive assays. You can use the same sensor peptide sequence in the time-resolved fluorescence, the PhosphoSens red format. That's shown with the histogram, where you see signal to background levels of 7 to 1 and Z primes of 0.94. And this is a particular target that actually was then part of an HTS screen with one of our clients. So we have had many groups come to us and say, with all the advantages of the PhosphoSens platform, you should really build a profiling service. And that's exactly what we've done. So we currently have 376 wild-type kinases in our large panel kinome profiling service. All of the spheres in blue or purple are the assays that we have in place. The key advantages really allow strong correlation with cell-based assays not seen with any other platforms. It includes the ability to run all of these in a continuous format, so we have greater accuracy and precision, and we have key information then for enzyme and drug performance, including the kinetics of time-dependent inhibition that we can see while running compounds in our profiling service. We can run this at high ATP, which is 1 millimolar, so physiological, but we can also run at low ATP to maximize inhibition and assess target scaffold reactivity. We run physiological levels of magnesium, manganese, and calcium. We again use physiologically relevant substrate sequences, so peptide sequences derived from appropriate substrates, with the minimum change in order to be able to see the appropriate optimized performance of a target kinase. We screen kinases from multiple suppliers so that this creates an optimized panel for the best results. And all of these assays are run on a single platform, so there's no sort of deconvoluting to understand whether a particular signal in one assay is equivalent to the signal seen with another assay format. Everything is on one platform, which is the PhosphoSens Kinetic format. And we're adding then new assays continuously. So just to show you then some of the results. So on the left is screening with staurosporin, a very well-known generic kinase inhibitor. We screen all of these at 1 micromolar and with 1 millimolar ATP. And so the staurosporin appropriately shows many kinases lighting up. If you do this at low ATP, you see almost everything lighting up. In the center is BI4020, which is a reference compound for EGF receptor. And yes, we see EGF receptor in HER2, but we also see many off-target hits. And we are routinely running reference compounds in our profiling service, and this is very common. On the far right, however, is an example of showing great selectivity, in this case for HER2, with one of our clients' compounds. And so clients are working with us as part of their SAR program. We're routinely running profiling, and they're able to then to modify the compound to give maximum selectivity for the target of interest. We can then look at all of the hits. We can scale these up in terms of doing individual IC50s to report out the potencies. And again, as I mentioned, we can flag time-dependent inhibition if we were to see this with, for example, covalent compounds. We can then take compounds and do residence time analysis. So we can do this a couple of different ways, either by looking at the forward progress curves or by doing a jump dilution method. The jump dilution method is shown here, where you take a particular target, you incubate it with 10 times the IC50 value, you do a pre-incubation for 1 hour, and then you do a 100-fold jump dilution so that any of the compound that comes off of the target can't rebind. And so what you're looking at then on these curves on the left, these are progress curves showing either the untreated enzyme in blue, and this is for the EGF receptor triple mutant. So you see a nice linear progress curve. And then with the red and the yellow, we're looking at 2 reversible compounds. One has a very short residence time, the red curve. So as the compound dissociates, it very quickly then regains almost full activity. The yellow has a longer residence time, and so you can see that characteristic arc. But at 60 minutes or beyond, it's now regained then the same reaction rate that you're seeing with the untreated control. So in contrast, looking at the green and the purple progress curves, what those are 2 different covalent inhibitors, and so you see much longer residence time. So that one expects, but we can also see really long residence times, even with reversible compounds. And so that example is shown on the far right, where a client sent us 30 different compounds. They all, if you look at the blue arrow, all of those red dots, right, are compounds that have a similar IC50 value. But if you then look at residence time, they differ by a huge order of magnitude, which is from 30 seconds to 2 days. So really dramatic, and this is with reversible compounds. So when we're looking at covalent compounds, the way you want to analyze them is by reporting a Kinact over Ki value. And this is because covalent compounds demonstrate time-dependent inhibition. And so if you look at the progress curve shown on the left, you can see, especially at higher drug concentrations, that there's an initial reaction rate, and then there's a final reaction rate. And in many cases, the final reaction rate can actually be 0. And so the way to analyze these is to then run this kind of 24-point titration of your compound, and then do a global fit across all of the data across the time course. And this is really important so that it allows you then to estimate the initial reaction rate, the K observed value, and the final reaction rate. And we do a least sum of squares determination using our automated data analysis templates so that we can determine whether it's 1-step or 2-step, and we determine then the Kinact over Ki value. If it's a 2-step, we also then are able to report the individual Kinact and Ki values. We provide the IC50 dose response curve and the Kops versus I plot as shown below. But we only provide these as a visual, and they really should not be used to actually direct your structure activity relationships. You really want to use the Kinact over Ki value. Again, if you look across those progress curves, if you did IC50 determinations, the question would be, at what time point do you do those? And so the IC50 value is going to continually change over time. The Kinact over Ki is powerful because it actually assesses that overall time course for each concentration of inhibitor. So to illustrate this, if you look on the right, this is a plot of the log IC50 versus the log Kinact over Ki. And each one of these red circles shows a series of compounds that have very similar IC50 that are then differentiated using the Kinact over Ki value. So again, very powerful. And we've had clients then send us data like this, where they'll say, wow, your functional biochemical assay giving us Kinact over Ki values correlates really nicely with our cell-based cytotoxicity assays, in this case, using cell titer glow to look at overall viability. This is for 2 different kinases that are related. And again, you see really nice correlation. So what it tells you is that you can spend a lot more time and less cost to optimize your compounds in a biochemical format, knowing that this will then play out in a positive way in your cell-based and ultimately in your clinical trials. So I mentioned in the beginning that we're very keen on developing selective sensor peptides so that one can look at crude cell or tissue lysates. And this is very important because we do so much with kinases or phosphatases as recombinant proteins, in a well. But you really want to understand also how the kinase is behaving with respect to individual complexes that might be formed in a live cell. And so what we can do with selective sensor peptides is to then make crude cell lysates or tissue lysates, either from appropriate cell models or, in this case, from matched tissue samples from the National Tissue Database. You then make those lysates. You can then do the same add and read format in any type of plate, and get very quantitative then measurements of activity of, in this case, on the upper right, showing a cell model for breast cancer progression, where as the cells go from a transformed, immortalized cell line all the way through to a completely malignant and 100% xenograft efficiency strain, you then see certain pathways become activated. And in this case, what it shows is that P38 alpha and beta are potently upregulated then in terms of their activity. In the bottom, same thing, looking at either cancerous versus normal from an individual patient. And what you see, again, is P38 alpha being upregulated in that cancer tissue. So very powerful. And we have increasing requests then to optimize some of our existing sensor peptides so that we can make them more selective to be able to do these kinds of applications and ultimately take those assays forward into the clinic to be able to use them as a biomarker. So just to summarize then, a lot of advantages then for the PhosphoSens platform. It is sensitive, continuous. It is a direct measure of activity. We use physiologically relevant sequences, and it gives you then a progress curve in every well so that you learn more about target enzymology as well as drug functional properties. Because one uses initial reaction rates, we get very high accuracy and precision. So Z primes of 0.7 are very common. As I showed you, we can get Z primes up at 0.8 or 0.9. All of our assays are optimized for appropriate enzyme conditions. You can use any ATP concentration, so that can be at the ATP Km, or it can be 1 to 2 millimolar, which is physiological. And you can use these assays with any cell type, sample type, as long as the sensor is selective in terms of being able to detect activity in a crude cell or tissue lysate. Both assay formats correct for compound autofluorescence. So in the Kinetic format, the autofluorescence won't change over time, so you can just subtract that as a blank. With the PhosphoSens red format, you're using europium and a time-resolved fluorescence readout. So you have a wavelength for emission of greater than 615, and therefore, along with the time-resolved fluorescence aspect, you are not seeing any contribution then from autofluorescent compounds. We've built then a rapid and predictable development process, and we have over 30,000 sensor peptides that we use as a panel to routinely go back to for individual branches of the kinome to develop new assays. We have rigorous manufacturing process in place, and so we have excellent lot-to-lot consistency. And the platform works then across the entire drug development process so that one can do PhosphoSens red initially with high throughput screens. One can use the Kinetic format for upfront drug target validation. One then goes downstream from HTS or SAR, where you're then looking at lead identification and optimization, and then ultimately into cell-based formats where one can do crude cell lysate formats using the PhosphoSens lysate selective sensor peptides. So being able to use the whole platform across the complete process accelerates drug development and improves the outcomes. So again, as I mentioned in the beginning, we have over 400 assays now as catalog products offered as kits and bulk sensor. The kits allow you to immediately just assess then how the assay works in your hands with your target enzyme, but you can then convert to purchasing bulk sensor to be able to get much greater value. We offer custom assay development under a master service agreement, 98% success rate, 3 milestones. We've done this systematically, even for really difficult targets. And then our custom testing services are highly interactive and have rapid turnaround times so that we can work with you even for SAR for within 5 days to provide you then data so that you can incorporate that into the next round of chemical synthesis. We're doing this routinely with many companies. Again, these assays use optimum enzyme conditions, ATP concentration of your choice, and the kinetic assessments then allow more information with higher accuracy and precision that we can apply to profiling, IC50 determinations, K inactive or Ki, residence time, mechanism of action, and EC50 studies to look at activators. So many advantages that really enable optimized and informative development and drug analysis.
Thank you very much, Eric. For those of you watching, if you had any questions or thoughts to share, please reach out to us at hello@assayquant.com. We also have a bonus Q&A directly below this video where our scientific team asked Eric great frequently asked questions by our customers. Just scroll down and hit play to hear the Q&A session. We look forward to hearing from you.